Hello, everyone. Welcome again to day two of the Water Summer School. Uh, with that, let me introduce today's speaker, Danyka Byrnes. Hopefully I'm pronouncing it correctly this time. She's going to talk about plotting your course with effective data exploration. And Danyka is a PhD candidate at University of Waterloo, Canada. If I'm not wrong, under Dr. Nandita Basu, and her research focuses on leveraging data to analyze anthropogenic impacts on water resources. Specifically, she's looking into the effects of climate, land use, and management practices on water quality issues. And I have known her for her leadership in AGU H3S. So without further ado, I would like to invite Danica. Let's welcome her. So Danica, you can go ahead and share screen.
Thank you so much. Okay. That should be, y'all should be seeing it, seeing my screen now. Thank you so much for inviting me today. I, spend a lot of time in looking at different forms and just different data sets that it's great to finally be able to have an outlet to talk about it that's not specific to kind of my research. So today I'm going to be kind of talking about data exploration, and I think it is frequently an overlooked part of research, and specifically when we do data-driven science. I think when we think about data science, a lot of the conversation revolves around all of the machine learning models that we use, the sophistication of it, whether we're using XYZ hyperparameters. But when it comes down to it, the data is arguably one of the most important parts of the model. So while good model architecture is important, we are able to really enhance our workflow and just like have better understanding of our research questions and just the process that we're trying to describe if we take time to properly look at the data. So kind of my talk and this workshop really aims to bring in some of the focus into the underappreciated part of data exploration. And then so I'm hoping that through what I talk about, as well as in the workbook later, I can help you gain some intuition or just some approaches that you could use to gain intuition about your data. So then you could answer the right questions, you could generate the right models or use the right models and generate the right hypotheses. So, starting out with who am I to be speaking on this topic? I'm a PhD candidate at the University of Waterloo. I'm in my 5th year. I should be defending in a matter of 4 months, if all goes well. My background is in environmental engineering, and having started out, in environmental engineering, we spend a lot of time coding, but often the data sets that we get when we're doing this, they're pre-cleaned because, the effort that goes into the data, kind of actually cleaning the data set, is extensive. So, when I was doing my coding exercises or kind of generating models, a lot of it was with kind of this perfect data. And we also see this when in tutorials that you might have done in when you're trying to learn any sort of machine learning approach. The data sets that you're given are usually very clean. They have a set amount of features that or attributes that they're given to you. So you kind of miss out on the steps or the important steps of kind of getting this raw data that is just riddled with errors, and then also not really knowing what to do with it. So the umbrella that I'm a hydrologist. I do biogeochemistry. I work on water quality, but those are kind of like the topical parts of my research. But what I really do is data analytics and data science. I use large data sets to be able to kind of glean different insights and just be able to kind of understand how the watershed is processing nutrients using kind of like data-driven approaches. And so mostly what I'm trying to answer is how do nitrogen inputs impact water quality or river loads? And we'll dive into the kind of the specifics of that in a bit. A large part of what I've done is data set creation. Me and my colleagues have put together these extensive data sets to be able to better answer that question, because there was an absence of data available to us. I take this data and I use machine learning models to be able to kind of glean insights and maybe sometimes even generate more data, so that I could finally answer questions that we want to ask, that we can't ask because we can't ask right now because the data does not exist. And then ultimately, kind of taking these data-driven approaches to be able to just taking data and trying to understand what's happening. So I just want to take a second to ask the audience, or you guys, what do you feel like your formal education provided enough practical skill for data management or data science? I assume some of you maybe are kind of more in a data science role, or kind of in a data science program. So maybe some of you are yes, but if you kind of have a non-traditional background, do you feel like that you have adequate training for it? I'll just take a second. Yeah, so we have one no. Yeah, I mean, same, very much same. Also, very self-taught. Yeah, and I think, Joshua, you bring up a great point. People who might have had some data science training, or even like statistics classes that have data science, machine learning models. Sometimes it's also not always the most applied, right? You're using the spam like data sets and everything, and it's not applied to either, like, your area of expertise. For me, it'd be earth science. So I think that, and I'm making a generalization here, but I think a lot of people here are in kind of the umbrella of earth science, and we don't have that formal training, and so a lot of the stuff that we learn is self-taught, which is great. It's adequate for what we do, but we end up kind of missing out on some really really key important facts, or kind of skills and approaches that, or just just general understanding that we might not have. But I'm kind of here to say that that's fine. That's definitely not a concern. I think that I see a lot of people in my field that are self-taught, and they also have that. But what's kind of makes them stand apart from other people that might have the formal training, is that they also have a domain knowledge. Anyway, so I'm just here to say, me too. I don't have formal training. Everything I've done is self-taught, but that's okay. So, like I said, I'm not an expert. If you have preferred approaches, or if you hear something that I say and it's like, oh yeah, I do it a different way, I encourage you to put in the chat. I think that we all have things that we know, approaches that we like, and the joy about kind of these groups getting together is that we can share these resources. And I have my knowledge, my path that I've taken, which I'm trying to share with you today, but there's also many different paths. Okay, so I want to kind of bring this kind of talk about how I brand myself, and I want to kind of put it into like a kind of two different ways to brand yourself and why they're both important. And the reason why I bring this up is because I think that as kind of people doing applied data science, it's important to kind of think about how you communicate who you are. So, my domain focus, I'm talking to other water scientists. I say I'm an eco-hydrologist. But if I'm trying to focus on a more skill focused, if I'm talking to someone about my skills, I will say I'm a data scientist, because that's fundamentally what I do. When I say eco-hydrologist, like very few people are going to know what that means. So it's not overly useful. I study how, my domain focused introduction is I study how nutrients move through the subsurface and impact water quality today and tomorrow. But I'm talking to someone who's more interested in my skills. I say I use data-driven approaches to answer relevant questions on how humans have impact, or the impacts of humans on the environment, right? So again, that's skill focused kind of perspective. And then, I use modeling and data to understand the fate of nutrients. And then my skill focus kind of introduction is I use disparate data and machine learning models to generate high value data products. So again, why I bring this up is the way that you kind of pitch yourself when you're trying to find kind of your niche, where you want to brand yourself, what you want people to know you as, kind of really depends on what you want to sell. And I think for maybe a lot of people like us, focusing on the skill focus approach can really put you maybe more in that data science domain. But you still need that kind of that domain focused introduction as well, because it's important. It gives a lot of context to what you kind of bring to the table with data science, or when you kind of do data science, the context you bring to the table. So kind of learning to brand yourself both ways. And this is how I brand myself. Okay, so moving on to the goal of the workshop, I really want to go through kind of 4 main things. We're going to start by talking about approaches to finding data. And I think that we all have this is kind of when I say I want y'all to talk about approaches that you use. This is one of the areas that I'm sure that we all have a million different, we just have many different approaches or ways that we all look for data. So I encourage you to put it in the chat if you have something else to add that I didn't mention. An appreciation for the complexity of cleaning data sets. We don't go extensively. None of the workshops are going to be about data cleaning because it is a little boring, but it is incredibly important and also extremely time consuming. Approaches for effective data exploration for insights. And this is what we're going to focus on in the coding workbook. And my intention is just to give you a few tools that you can use when you kind of have this fresh new data set that you just cleaned and it's overwhelming because there's a lot. What can you do to be able to kind of start working towards developing a hypothesis or kind of knowing what your data can tell you? And then lastly, kind of the importance of that domain knowledge in data science. A small anecdote that I'll use is I was talking to someone who ran a team in Environment Canada. So that is like our EPA. And she was doing water research, but she only hired computer scientists. And, when we met with her, it was obvious that we brought it was me and a few colleagues. We brought so much domain knowledge to the conversation that the other computer scientists didn't know. And that's not to say one is better than the other, but it's just, having that background, having that ability to look at environmental data and understand it because you understand the processes that drive it, kind of adds an extra layer of just an extra layer of insight and just kind of something else to bring to the table. Okay, so I'm going to kind of quickly go through my research. I know that, not everyone's interested in water quality, so I tried to keep it kind of only the kind of 1,000 feet level kind of introduction of my work. But so, my main focus is trying to understand how nitrogen from humans kind of moves through the landscape. And there's really kind of a few main sources of nitrogen or nutrients that I look at. And it's mostly, a lot from fertilizer. There is biological nitrogen fixation. There's human sewage, atmospheric deposition. So from rain, you'll get nitrogen that goes onto the ground, onto the watershed or the earth surface. And then livestock manure. So these are all what we call inputs into the watershed. And when we think about, or when we talk about kind of the pollution, it's these sources that we're most focused on. And so, as these go into the landscape, they move through either through runoff on the subsurface, or they move through the groundwater to go and pollute our rivers. And the time that it takes for kind of this nitrogen to go from the landscape to our rivers is not, it's not straightforward, right? The path it has to go through, especially when it goes through the subsurface, where it gets taken up by things and released, and goes through some chemical, transformations, can really take, maybe a year to like 30 years to get through the landscape. And we don't have a good understanding of kind of that kind of relationship between what goes in and then how it shows up, or when it shows up in the river. And so, this is an incredibly large issue. I, tried to actually, these two examples are highly relevant to those in Texas. I don't know if you are all sitting there, but the Gulf of Mexico every year for the last, I think, like at least 30 years have had just this ginormous algal or dead zone. And it's caused by kind of the mass amount of nutrients from the Mississippi River Basin going into the Gulf of Mexico. And essentially that just consumes all the oxygen in the water and creates kind of these areas in the water column, kind of in the water, that has no oxygen. And so fish can't breathe and they die. And that's obviously not that's not an outcome that we want. It's bad. It's bad for tourism. It's bad for fishing industry. It's also just bad for the environment. So, there's a lot of desire to kind of address these nutrients and understand what's going on so that we could kind of stop doing it, stop putting nitrogen in the Gulf of Mexico. We also see kind of these, with these feedlots across the US, this one particularly is in Texas, just mass amount of nutrients coming from livestock and their manure, and it goes into kind of water and then just produces these, giant algal mats, algal blooms. And, that water has to go somewhere. So either it goes through the ground, it goes through the subsurface through groundwater, or it goes to a river. And then, either we drink it or or it goes into our rivers and lakes and causes algal blooms, and kills fish. So, with this idea, so let's say, we're trying to mitigate this issue. So we're saying, okay, we want to kind of reduce how much nitrogen is going into our rivers. So the obvious answer, and what a lot of the research has been kind of focused on in the last 30 years, is if we kind of put less nutrients in the landscape, so that manure, that fertilizer, that atmospheric deposition, if we put less of that, then what's in the river is also going to decrease, right? So that red dot, if we're saying we want to decrease that net nitrogen input, so how much we put into the landscape, it should decrease how much is in the river. It should decrease that nitrogen load. But that's really not what happens. So we see these effects, and they're called hysteresis effects. What drives them? Well, maybe I'll talk briefly about what drives them, but essentially what we end up seeing is, if you look at the panel on the right-hand side, in the 1970s, net nitrogen inputs it kind of continues to go up, and it goes flat. And then at a certain point, our net nitrogen inputs change. So, or rather, our despite kind of net nitrogen inputs kind of being the same, we see our load decreasing. So, so there's kind of like this disconnect. So we're doing the same thing. We're putting the same amount of nitrogen in our watersheds and on the landscape, but we're seeing load respond in a weird way. Similarly, if we see on the left-hand side panel, we see inputs go up, and that at a certain point they flatten out in the 1990s. But even though there's kind of the same amount of nitrogen going in, our load continues to increase. So what I'm trying to kind of get at here is that there's kind of this nonlinear response of kind of what we're putting in and what's coming out in the rivers. And this is just to highlight that we really don't have a good grasp on what drives these relationships and why they're so different across space and time. And so we've dug into a little bit of this in previous papers, and we'll kind of get into a little bit of that in a bit. But so when we try to understand what drives that relationship, what makes kind of this decoupling or this non-linear relationship between what goes into the landscape and what comes out, we're kind of focused on stuff called this term called legacies. And really it's this pool of nutrients that that is kind of just, it's hanging out in the landscape. And then so no matter what you do, if you start decreasing how much nitrogen you put into the watershed, because there's this pool of nutrients kind of accumulated from decades of just adding fertilizer and manure to the landscape, whatever we do today is not really going to matter until this kind of pool of nutrients gets depleted. So this kind of creates this constant, persistent source of nitrogen, or these legacy sources. It's like this kind of persistent source of nitrogen in the landscape that just kind of makes these relationships between what goes in and what comes out really challenging to understand. And so when we look at a watershed, you know, the biogeochemical legacies are going to be kind of nitrogen stored in the soil. Hydrological legacies is the nitrogen stored in the groundwater. And then kind of where the nutrients are in the landscape is really dependent on, you know, kind of soil type, how much carbon is there, what has kind of land management, what have people done to the land, and what's the climate like. And similarly, if with hydrological legacies, what kind of drives how much nitrogen is in that pool is, again, climate, soil type, drainage system. So what I mean by that is whether people have put in kind of these giant tubes in the subsurface to drain kind of their crop fields to just kind of remove water from the crop fields. And then also if there's irrigation. The last 2 points are not so important if you don't know what they are. It's just to demonstrate that there are different things that are driving it. Again, we don't have a good idea of what's causing it. But what we end up seeing, so when we look at the outlet of, this is of the Mississippi River Basin, so the Gulf of Mexico, where that big algal bloom is, what we end up seeing is that the nitrogen, if we were to kind of take like a grab of water from the outlet of the Mississippi River Basin, we would see that there's nitrogen of all different ages, right? There's nitrogen that is over 30 years old. There's nitrogen that's, you know, under 5 years old. So trying to manage trying to reduce the nitrogen that's coming out at the outlet of the watershed is requires kind of addressing all these different pools of nitrogen and kind of, you know, reducing what goes in now, but then also addressing kind of this old 30-year-old nitrogen in the landscape. And then, you know, what drives water quality is a function of today and yesterday's nitrogen use. And so this is kind of like the crux of my research, is what drives changes in nitrogen loads. So this is the context at which I approach all my kind of data science activities, kind of data analytics, is trying to understand, have a better understanding of what's going on in the subsurface. And I mostly do this through kind of data analytics, just seeing how much is going in, what's coming out. And I'll talk about a few specific examples of the 3 research projects I have on my plate at the end of the presentation. Okay, so with this context, you know, where do I find data? And I think that this is very applicable if you don't do water quality, if you don't kind of care about water quality, if you're in a completely different domain. I think a lot of these databases are still relevant to you. I think I find a lot of data kind of in journals, right? The last, I don't know, maybe 5 years, we've seen kind of this exploding of journals that are dedicated to data set kind of documentation and publications of just data sets, which is incredible. And there's so much data right now. Just going through scientific data, the journal, you know, if you wanted to just do a project on kind of like a random topic, you would have just too much to handle if you tried to go through scientific data. And it's very broad as to what topics they cover. You know, Google has a data set search function. I think that maybe in 5 or 10 years, or maybe 5 years, it'll start being good. But I find the indexing is not great. Really hard to find what you need there. But it's always nice to kind of check out what they have indexed on Google. And then there's repositories, and I think that, when kind of in the process, when you're trying to kind of at least build up this kind of like database domain, kind of know what kind of data is out there, turning to these, bit overwhelming, but kind of just kind of browsing through what data is available can be a pretty good use of your time. And then lastly, like if you are in the water quality field, the USGS has incredible data, and they have extremely good metadata. And of course, there's NASA. I've never used NASA data, but I'm sure a few of you have. So when you kind of like, if you're getting started, or if you're even still in here, knowing what players are kind of in your domain, so knowing where to go for data, where are the best places to go for data, who are the authors of the data sets that you're looking for, can be extremely, like, just knowing that. So then periodically you can go and scour different, like all of these resources, and find new data sets that come out. This will put you on, like, just, knowing what data is available is helpful when you're trying to talk about research projects, when you're trying to think of new projects to do, when you're kind of struggling with something in your current research project, and you need another data layer. Knowing what's available it'll give you so much of a step up. And then you also get to talk to your colleagues about what data is available, and you kind of just become the data person, which is always a nice place to be. Again, going back to branding, it's kind of important that people know you as a data science or data person if you kind of want to be in that sphere. And then also I subscribe to emails from data papers. So with scientific data, I get a notification every week of what papers have come out. And in the end, there's a lot of wasted kind of like, of course, I'm not using most of the data that's coming out, but when I do see something I need, I'm able to document it. So I highly encourage y'all to do this, if you don't already, is just document your data or the data sets in some way. I started out using Google Docs, and it's extremely, kind of rudimentary, just kind of putting the data set names and keywords so I can control F it, and then URL. Do that if you're busy and you don't really kind of have the time to set up a huge database. But then, if you're able to, using something like Obsidian, where you're able to kind of make these, you can put in your data set information, and then you could also tag kind of what data is in there, the temporal resolution, the spatial resolution. You can really start kind of putting together networks of data sets. And then when it comes time to do a project, you're able to kind of quickly go like, oh, I need this, or, oh, I'm thinking I might need something about, let's say, human waste. Okay, let's go to my data set and figure out all the stuff that I have, and you have it all laid out. Again, because, if you're trying to make yourself like, or if you're trying to be in that data science space, having kind of this setup just makes it's extremely advantageous. And it like I said, it gives you a leg up on people in your field. So the next part you could also do is create your own data sets. And this is something that me and my colleagues have been doing for a few years now. I started this in my master's, and we're still kind of making upgrades to our data. And it's just a continuous process of kind of updating data sets. And what we generated was what we call the trend data set products. And essentially, they're just long-term nitrogen and phosphorus data sets. These spanned 88 years. Some of them are county scale, some of them are gridded, so the 250 meter scale, and they are across the US and Canada. There's, this step, this kind of data set creation is very cumbersome. It is a huge undertaking, but it could really help with kind of, visibility in your field. If you create a really good data set that people need, you do become a little bit more of a face that people recognize, because you're the person that generated this incredible data set. And then you just need to know kind of where the need is in your field, and then also how you can do it, and kind of the steps and the pitfalls that come with kind of whatever domain you're working in. So we created these data sets, and it's kind of this never-ending project, but it allows us to kind of be in the data space. So when you create your own data sets, of course, you can use things that are easily accessible, but there's also, I just kind of want to mention that we have, of course, a lot of data right now. Every day, a lot of data is being generated, but there's also some very good historical data sets that are not digitized, that are kind of, they are underutilized, forgotten about, and they hold a ton of information. So for example, for this trend products, we're using agricultural census data, and we're using data from the 1930s, and of course, much of that is not digitized. So being able to kind of take this data and digitize it, and then put together a data product with it, is just huge value added. And from this, with that proxy data, whenever you're thinking about creating a data set, you're probably never going to find the exact kind of data points you need. For example, for me, it'd be nitrogen, but I can get how many cows there are. And so if I think about, kind of when you're trying to think about data and creating data sets, you really want to be thinking about kind of what proxy data can I use? What can I use? What kind of data can I get? What models can I apply to be able to get the data product that I want? And then, yeah, so for cows, it's just like, how many cows there are. And then I could use a kind of an empirical model to put that, kind of translate that into how much manure is being created, or how much bushels of corn there is, and how much nitrogen that removes if there's X bushels of corn in one county. Okay, so yeah, so trying to rush through this piece because, of course, it's an incredibly important part of kind of this being in data, especially in like research data, and it all kind of falls under the domain of data exploration. But kind of moving on from that, I think that I'm sure most people are very familiar with the kind of the FAIR principles, so I don't really want to get into this deeply, but it's worth mentioning. If you're creating your own data sets, or even, if you're creating your own data sets, or kind of even publishing modeled results, you definitely want to be looking at these FAIR principles. So when you're approaching this and you're creating your own data sets, you definitely want to maintain a log of all your data sources. And I say this mostly as a warning, because I think that hopefully you guys aren't like me, but I have a tendency to work really fast, and then kind of skip steps of kind of proper data management at the start. And it really impacts me at the end, where then I have to kind of go through all my kind of data and sources and kind of patch together where I got stuff. It's doable. I keep good enough records to do that. But if you just do this at the start, much easier, and it'll just save you a huge headache. When you're kind of going through this data set creation, keep a record, don't manually change data, have code that changes your data, but keep a record of how all the data is transformed, including the cleaning and aggregation steps, right? Everything should be documented, because if there's anything I learned in the last 5 years, is that I do not remember what I've done 3 months ago. And oftentimes it's kind of it's a time sink if you have to go back and figure out, like, how did I aggregate this data? Was it mean, median, blah, blah, blah. So remember, kind of keeping a record of how you do this, whether that's through Jupyter Notebooks or through code, or just a really detailed method section, will just save you in the long run. An important step that I think is also underappreciated is standardizing validation rules. So when you create these data sets, you want to have some sort of kind of consistent way that you check your data for accuracy and for errors, because in the kind of, you're very likely going to make mistakes, and the only way to catch them is if you kind of have standardized approaches and you just plot your data every which way to make sure that it all looks kind of as it should, no matter how you plot it. Again, keeping a data dictionary for all the variables in your data set. This will help you when you're building your metadata. And then creating kind of a template or standardizing data to ensure uniformity, so it's machine readable. Of course, standardizing zeros and no data. I've worked with data sets where people, they have not done that, and you lose a lot of kind of resolution of the code when you don't know what's no data and what's a zero. So it's underappreciated, and just, following these simple steps will save you time, will make your data set better, and will just be generally less of a headache. So I want to take a second just to talk about metadata. What time is it? Sounds extremely boring, but I think that we only got a few minutes left for this part, but I wanted to try to kind of do an exercise with you guys, and so I put two metadata files in there, and you have a great opportunity to kind of roast me, because one of them is mine from my first data set that I published, and then it's being compared against a USGS metadata. The USGS has done a lot of work in trying and like kind of standardizing the metadata approach. And the details they have in there is incredible, like it's just incredible. They include everything, how they transform their data. So I highly suggest, if you're going to be creating data sets and you want a template to kind of model your metadata off of, look at the USGS on how they do it. And it'll just kind of help you know what you don't know when you're kind of when you're trying to put together this metadata, so that your data set is usable, and you're not just left with kind of a data set that no one understands. Wait, maybe we'll get to this. Maybe we'll try to get to this step later, to just kind of just take a look at those two. If you're able to take a look at those two metadata examples that I have, figure out what you like and don't like about them. And then if we have time at the end of the session, we'll circle back to this, and we'll take a look, and we'll just chat about the things I did wrong when I didn't know what I was doing. Okay, so data cleaning, again, underappreciated step, but this is where I spend roughly 80% of my time, kind of as a data person, data scientist. My research projects, unfortunately, is if I were to just like plot out what time was spent on what, probably 20% of my time is actually spent on analysis and kind of like hypothesis testing and kind of just figuring out what I want to say in my discussion. And then 80% of it is on data cleaning, which is not very glamorous, but it is arguably the most important part of the whole process. It can be very iterative, and it could span many months. And like I said, you want to plan to forget what the details are of your workflow. I promise you, most of you will not remember that one number that you changed manually in an Excel spreadsheet. So never do things by hand. Keeping track of your steps, i.e. through code or just really good notebook notes, will just be crucial for methods. And there's nothing worse than trying to figure out what you did a year after you've done it, when you're trying to write your methods section of your paper. So I guess I kind of gave away how much time I spend on data cleaning, but just for those who might work on data, how much time, if you're working on a data set right now, what's your average time spent on kind of data cleaning on your projects? Just post it in the chat, please. Yeah, I think a couple of months. I mean, weeks, that's fantastic. Yeah, it absolutely depends on the data size. Some data sets are, much smaller. You could handle it a lot better. Yeah, I think I'm now 7 years into the trend data products, and arguably I'm still doing data cleaning. There's a lot of times where I find numbers that are slightly off, and that's not necessarily always my fault, but there's things that always kind of show up. So it's always important to kind of just keep it in mind that, the data cleaning step, least glamorous, but the most important. We're kind of part data scientists, part just janitors.
Also, Danica, while people are answering that, maybe we can take a couple of questions.
Sure.
Um, one of them being my personal interest is I remember when we talk about nitrogen, and specifically if you're talking about fertilizers or especially in croplands, it depends on the kind of cultivation as well. For example, in my part of the world, uh, we have rice cultivation where you
Hmm.
have ponded water, right? And then the amount of denitrification happens changes based on your land management. So my question was, how is it being tracked? For example, I mean, you gave an example of how much nitrogen is being put in is sort of calculated approximately by using some other methods,
Hmm.
crop residues or other things. But how much has been lost uh, due to denitrification or maybe other processes? How is that accounted for?
Yeah, so that is actually the $1 million question. Denitrification is one of the most challenging processes to estimate because it is incredibly temporally and spatially just like variable. So frankly, we don't know how much is being lost to denitrification on a very large scale. There's estimates, and part of actually the research, like my last chapter of my thesis, is trying to get at those numbers a little bit better, trying to understand, okay, we're losing this much to crop uptake, this much to river loads, and then trying to back calculate. We're going to try to back calculate how much is lost to denitrification, but ultimately on a large scale, we don't have a good idea of how much is being lost. So I know that's kind of a non-answer, but yeah, it's like very much still up in the air.
I was assuming that because, and also, are there any sensors available to sort of compute how much is being lost uh, due to denitrification, or how do people generally quantify that? If there's any mechanism, or is it all
Mhm.
backtrack?
Yeah, so denitrification at the field scale will be measured through kind of field instruments, very small pilot, like kind of very small studies of just measuring how much, like what nitrogen's being lost. So you can track how much nitrogen's coming from the ground. But so that's at the small scale. At the larger scale, there's not really anything that quantifies it except for models. But even models, because there's not a good way to constrain those values, they are still just kind of estimates. There's been some budgets, like, using nitrogen budgets is one way to try to kind of guess at how much is being lost through denitrification. But in the end, it is still kind of like, okay, and then we're assuming the rest is lost to denitrification. So it's not it's not a measurement of it. It's like okay, we think we accounted for everything else, therefore this is probably what's lost to denitrification. And there aren't that many data sets that kind of that quantify that, especially at the large scale. So a very large data gap in case someone wants to take that or take on that undertaking, take on that data set.
Yeah. And one more comment, maybe. So the topics you're touching upon, for example, is so relevant right now that because of it, open science paradigm is pushed so much. I think you have to make your data available. You have to make your codes
Mhm.
available, and I think that transition has to happen. And that, yeah, I just wanted to mention that.
Yeah, no, I think you're right. And I think, again, because most of us are coming in without that training, I feel like a lot of times we're left to figure it out for ourselves. Like, people talk about what open data is, and, we might have a general knowledge and like, okay, we need metadata, but then there's no one really telling us, okay, no, this is what you need. This is how you do proper database management. And there's this huge gap that we all find ourselves in because we're going into data, we're using big data, or at least like a lot of data, but then we're kind of figuring it out as we go, which it's unfortunate, and it leads to a lot of sloppiness, and including myself, sloppy data sets, and you kind of look back and you're like, oh, I could have done that better. I should have documented this differently. But like, I really appreciate this kind of drive towards open science. But there really isn't that much infrastructure to help us, especially students who are trying to learn, with PIs who maybe didn't kind of also do data because they find themselves in this new kind of data age now. We're left to figure it out for ourselves. So I don't know. I don't have a good answer to that, but I think that it is relevant, but it's also really hard to that, like, we're kind of just being pushed into the deep end. Yeah, so I was trying to read the one chat, but I guess y'all are talking about nitrogen collection. Love it. Or denitrification. Okay, so I'll just kind of move along. We have 10 minutes left, so I'm going to try to just kind of move through these pieces a little bit more quickly. When we talk about data cleaning, kind of removing irrelevant data, removing structural errors, making sure that things have the same units, checking quality flags or provisional data. And Tamlin from UNC, who is kind of one of the lead researchers, or kind of lead researchers on the SWAT satellite. He had this great data chat, and he said, my recommendation when users start working with these data, or any data, is to go and download the product description documents and look through it. Again, incredibly boring, not nearly as fun as playing with the data, but kind of doing these steps on the front end helps with kind of like not pulling out your hair, or not like doing a bunch of analysis and realizing that you understood the data improperly, or you didn't consider the different units, or trends that you expect to see aren't showing up. Why? So just keep that in mind. Always kind of do yourself a favor, do the work in the front end, and then you can just kind of play with the data later and play with clean data. Standardizing, removing duplicates, handling outliers and missing values. This is definitely more of an art than a science. I don't have a good kind of straightforward approaches. This is very much a domain specific approach. For example, looking at kind of what distributions you have, and kind of, is a value a reasonable value, even though it's 3 standard deviations away or 4 standard deviations away. Kind of understanding this. And this is where the domain knowledge becomes incredibly important, because you can make these decisions because you know the data you're working with. It's iterative approach, right? You might think you cleaned everything, you go to plot something, a data point is kind of wonky, you got to circle back to it. So again, this is why it takes months. Whenever you're kind of dealing with cleaning data, I always like to kind of say, embrace skepticism. If a data point looks funny, go and investigate it, because it's at those points where you start pulling on threads. Even if it's a data set that's supposed to be already cleaned, when you start pulling on those threads of weird looking data, you usually find issues at the end of the thread. And then if you don't, at least you spend this time getting a gut feeling for your data. Again, like I was saying, what's considered an outlier, it's domain specific. It depends on the distribution of your data, depends on your research question. If you're worried about flood flows, you're not going to care if there's kind of like, issues with low flow values. But if you're really worried about low flow values, and like water gauge got knocked out because there was a huge flood, in 1997, probably doesn't matter. So again, very specific. It's more of an art than a science. Data validation, this is just, I guess I could just, this warning, what I always like to do is plot your data in just every single possible way you could think of, and you'll find some mistakes. And if you don't, chances are when someone goes to use your data, they'll find the mistakes. So it's kind of better if you find it first, but letting other people play with your data is also a great way to find where things are kind of maybe a bit wonky. An incredibly kind of nice approach that is starting to be talked about right now is this split apply combine. And it's just this kind of like, data cleaning, or just this data processing approach that allows you to kind of with ease split up your data, apply different functions to it, and just look at it in many different ways. So this is relevant for data cleaning, and it's also something that we use a lot in the code book that we're going to work on in a bit. I'll skip through this because I'll explain it during the code book session. And if when we talk about it, if it's interesting, there's a whole paper on it from Hadley Wickham, which if that name sounds familiar, it's because he's big in the R community. Again, just trying to make sure that I give you some warnings that I had to learn the hard way. You will make mistakes. they're inevitable. Everyone makes mistakes. I haven't found mistakes in published data, and some from some really large agencies that just have extremely wonky values. No one talks about their mistakes, but we all make them. And it would really be great if we started to kind of get into a culture that we could talk about our mistakes openly. Oh, oops, I published a data set and like, oh, there was a few numbers that were wrong, but it's okay. I re-uploaded it and it's good now. So whenever, if you do find yourself in that position where you've made a mistake, I'm here to tell you that we all have as well. And, don't beat yourself up. Don't beat yourself up over it. It's part of the process. We're dealing with a lot of data. Okay, so that's kind of like the precursor / it's still part of the data exploration process, but it's not really the fun part where you get to look for insights, where you're starting to think about what kind of questions you want to ask, right? So, looking for outliers and errors, that's of course what we talked about, seeing if there's bias in the data. Data cleaning is obviously part of data exploration, building your gut feeling for the data. But the fun part is looking for trends and patterns, and that's what we're going to be doing in the workbook. I liken kind of data exploration to a meander in the forest, just meandering in the forest. You kind of have this like very loose path, where you want to go. You kind of, maybe you have general research area topic that you want to understand, but you don't know what you don't know about the data, right? You have to make sure that the data is without taking a look at your data and understanding what kind of the limitations of it are, you might not be able to really build up a kind of robust hypothesis or research question. So, embrace the meander in the forest, embrace the hike, because, it is a crucial step to understanding your data. And often it's the fun part where you get to discover certain kind of patterns, trends, and just, I don't know, play with some data. And then finally, we do hopefully get to the kind of data presentation part. And this is where it kind of is this neat little curated package, a beautiful story, but it's because we spent all this time walking around that we're able to do that like data presentation or the guided hike. It's because we took the time in the front end to kind of look through it all and figure out what's cool about our data. So I have 5 minutes before I want to jump to the code book. So I just want to talk a little bit about kind of the works that I have done with kind of using data analytics, and then some data science. So when we talk about nitrogen going into the landscape, we often refer to it as nitrogen surplus. Nitrogen surplus is just what goes in. So fertilizer, manure, biological nitrogen fixation, human waste, deposition, minus what's removed by crops. And essentially you're left with a magnitude of how much nitrogen is left over that's not productive, that could possibly go and pollute some water. There's of course the denitrification component, but because it's so hard to estimate, we kind of just group it in with the kind of excess nitrogen. And so with this, I was talking about the trend data sets earlier. This was the first, like kind of the first publication of it. And with this data set, we took the time to kind of understand how nitrogen sources and sinks, the sources being kind of what kind of the nitrogen going into the landscape, and sinks what's coming out, how have they changed throughout time and space. And so to be able to kind of get at that, we used k-means clustering, so a really simple machine learning algorithm. And essentially what it does is it groups together data points that are similar, right? So similar data points are going to be grouped together based on in X amount of clusters or K number of clusters. And then from there, you can figure out why are they similar. So I have this little GIF here shows kind of the general steps to k-means clustering. I'm just trying to wait for it to restart so I can talk through it. Right, so you have your data, you initialize your clusters, you take distance measurements for every point. And then every point is grouped together with other points that are very similar to it. And then the cluster center reinitializes, and then it takes measurements from each data point to every cluster center. And then until the cluster center stops moving, or at which point it stops moving, those are going to be the clusters. Those are like, okay, your error metric, your loss function is not going to get any better. Therefore, these are the clusters that, or these data points are the ones that are clustered together. And so like I said, the loss function is just the sum of distances between them. So it is a very, I'm trying to go quickly over this approach, but it is a very simple kind of machine learning, unsupervised. We're not telling it like, oh, these data points should be together because they're really similar. No, we're letting the data speak for itself. So we use that. Oh, I guess, and then, a step to be able to do that is data normalization. Again, more of an art than a science, right? You have to be able to kind of know your data to know which one's better. So the few that, that we can use is min-max normalization. Really bad with outliers because you're trying to squish everything between 0 and 1. So if you have an outlier, you're going to get just kind of this bimodal kind of big number, very small number, nothing in between. Feature clipping, you just take your values and you say, okay, everything above this number, smush that in. We're going to take these outliers and bring them in. Z-score normalization, it could be used for kind of not perfectly normally distributed data, but it is sensitive to outliers. And then there's log scaling. If you have data that's log distributed, considering log scaling your data for the normalization could be a good approach. And why do we want normalization? Well, if you remember, we're taking distance measures, right? So if you have a value that's, the magnitude is 20,000, and then you have another value that the, magnitude is like the max magnitude is 5, when it's trying to measure distances, it's obviously going to kind of favor the ones that are closer. So by putting everything on the same playing field or the same kind of scale, those distance measures are a lot more meaningful. So we'll go through this in the workbook because we use k-means clustering. And so with this, we're able to group together kind of different counties, and we're able to say, okay, these counties are the ones that have a lot of manure in it. These ones are the urban counties. These ones are minimally impacted, right? They don't have much inputs, and what's going into like the amount of nitrogen that's the surplus of nitrogen in these landscapes is really low. And we were able to kind of divvy up the whole United States based on kind of these 4 typologies. And what insights we get from that is that, especially looking at the crop dominated ones or the heavy like the counties that are extremely agriculturally driven, we're seeing a homogenization, right? So when humans convert, like they'll take this like, these three very different landscapes that look extremely different across space. We turn it into an agricultural field, and then we're getting this homogenization. So no matter, these 3 landscapes that were very different in the past now look the exact same. And this has huge implications for, biology, biodiversity, habitat, nutrients and everything. So, we were able to use kind of this historical data set that we created, cluster together, and gain insights about kind of what we're seeing across the US in terms of kind of, the landscape. I'll dive through this one quickly, and then we'll move on to the workbook. So another project that we worked on, this was using, again, that nitrogen surplus, so that trend data set, and then we paired it with water quality. And what we wanted to ask is how do inputs relate to outputs? So how does nitrogen inputs into the watershed relate to the outputs? And this was a project that actually the data exploration step was frankly many years long, and it was pretty onerous because the data was extremely messy. But we finally found something, and what we really were able to pull out of this from this kind of this black box data analytic approach is 3 patterns of how the nitrogen inputs or nitrogen surplus relate to nitrogen loads. And what we're able to understand from this is kind of, if we want to clean up, or if we want to reduce the amount of nitrogen that goes into our rivers, understanding how nitrogen is processed in certain watersheds is incredibly important. So kind of understanding where the watersheds fall in these 3 typologies can be really insightful to water managers. And so, the big crux of this, we did some feature engineering, and we calculated a hysteresis index. And so I'm just showing you a hysteresis loop here, and it's essentially through time, as surplus goes up, the way that the way that loads relate to surplus over time is different. So then, even though surplus is going down, it's not a linear relationship. It has this hysteresis loop. And so that really gives us, without going into too much detail about it, it gives us insight of how nitrogen is kind of moving through the landscape. And essentially what we can say is if there's a positive hysteresis index, or B, it's a kind of, looks a certain way, and we can kind of tell that it has a lagged effect. A negative one clockwise loop, it means that there's probably kind of wastewater treatment plant or some sort of nitrogen removal in the landscape. If you have further questions about this, we can dive into it a bit later. And then, with this, we're able to say, okay, so we have these watersheds that fall into these three patterns. What kind of watershed features are most common in these three typologies? And so what we found is in the green, kind of counterclockwise hysteresis loop, we found that there was a lot more tile drainage. So, this is just the process of installing tubes underneath the landscape to move water really quickly. And so, with this kind of data-driven approach, we're able to pair two data sets together, throw in some other watershed attributes, and really get a better understanding of how nitrogen moves in the landscape. So I'll skip the last kind of project that I talked about just for the sake of time, and we'll move on to the coding workbooks. So I have a metadata file in the input folder. If you're curious about what some of the variables mean, I definitely either ask me or look in the metadata. There's two workbooks. There's the data. There's the one that's like analysis, data exploration. That's what we're going to be doing today. If you want to go through some data cleaning steps, there's also another workbook that I put together. And you can work through that and just see what kind of, details I chose, or what approaches I chose to apply to my data cleaning steps, just to get kind of other insights on how other people do it. So with that, thank you for listening to me. I'll just acknowledge the people that have helped me along the way with this. And then, we'll move on to the coding part. Okay, I'll keep the Q&A open and also the chat. So if y'all have any questions, please feel free to post them. And if I don't know if people can unmute, but if you are able to unmute, please feel free to also just chime using audio as well. Okay, so like I said, there's the notebook that y'all can use to kind of do this along with me. If you find this is too simple because, it's data exploration, we're not kind of doing any heavy math or anything. If you kind of cruise through the notebook and you're like, that was easy, now I'm done. I have prompts at the bottom to do further data exploration that I haven't done in this notebook. So, feel free to do that if you want. All right, so I think in your notebooks, you don't have the path set up. I left it empty. So you should go ahead and run that before you try to run everything else, because you do need to load in your data. And all you want to do, you just want it to be mapping to your inputs folder, unless you changed around what kind of the format that I uploaded the folder in. Kind of a full path to the input folder should work just fine. Okay, so we're just gonna load in the data. All right, so the data that I put together for us today is concentration and discharge data. So concentration is just nitrogen. Discharge, of course, that's just another word for flow. And what we want to look at is the relationship between Q concentration, C concentration, and Q discharge, and what drives different patterns that we see. So very fundamentally just a kind of data exploration. What are we going to find? This relationship has been kind of explored a lot in literature because it is a very kind of, the relationship between Q and C is very insightful to how nitrogen or any kind of solute is moving through the landscape. And the relationship between flow, which, if you're a hydrologist, that flow can drive a lot of kind of solute or nutrient movement. There's some papers that I put there in case you're interested in yeah, you're interested in kind of reading more about it. But for now, we'll dive into a little bit what the relationship means a bit more. But if we were approaching this as people coming to this data set brand new, we would first have to kind of understand what data we're working with. So that's what we're going to do. We're going to try to, our first step is to understand our watersheds. So we're just loading in here some our watershed shape files, different boundary, kind of just watershed shapes and boundaries, mostly for plotting. Okay, so the data set we're using is called Gauges Watersheds, and it's a USGS kind of data set. What we want to do is, I think I'm using it. Oh, no. What we want to do is kind of first start off by plotting where our watersheds are. And if you've noticed, for this one, we filtered out a few of the shapes, the watersheds that we had in our data set. And what we did, this first step right here, what we did is with our concentration, our CQ data, we don't have data for every single watershed in that data set. So we just said, okay, of all the shape files that we have, what do we have data for? And so when we run, you should be able to run this, kind of create this map. And sometimes it takes a little while on my computer if it's being taxed. And what we'll end up seeing. Okay, so we have in the blue, we have the watersheds that are in our data set, and we can see that of all the watersheds in the gauges data set, we are missing a few of them, but generally we have really good coverage. So our CQ data covers a lot of the US, which is good. If we're trying to approach questions, if we're trying to ask questions about how concentration and discharge relate to each other over space, we don't want to have huge gaps in the spatial data set, because then we're starting to run into limitations, right? So kind of the first step that you might want to do is just understanding, okay, what data do I have? What's the coverage? What are we working with, right? So the number of watersheds in the data set is 2,594. If you've ever worked with concentration data, this is actually quite a bit. There is quite a bit of data in this because we're taking kind of we're not looking for time series. We're just taking kind of a watershed. It has one data point. Great, we're taking it. So our next question is, okay, how many data points do we have in each watershed? So, and I think, let me just flip back, make sure that I'm giving this to y'all. Yeah, so these few points, the starting points, just because they're kind of onerous plotting exercises, I just gave you the code. So what we want to see is how much data do we have, right? And across space and time. If we're trying to look at kind of how C and Q relate to one another, we want to make sure that, we're not just having one data point that's, or we're not putting a bunch of emphasis on one data point or on one watershed that has one data point and saying, trying to deduce really large trends from this. So what we generally see is there are a lot of watersheds that have a significant amount of data, over 100 data points. And throughout time, we also have a good amount of observations throughout the entire kind of time period. So that's great. That means that we have good spatial and temporal coverage. Again, we have good coverage, watersheds across the contiguous US. Although if we look at the map, we notice that, okay, it's a little bit more sparse in the West, which makes sense. There's a lot less rainfall there. We don't gauge watersheds as much, and we're less concerned about nitrogen in the Western US. Because a lot of the areas that we see water pollution are in the East, in Gulf of Mexico. So kind of Midwest, Eastern US is where we care more about it. So that makes sense. But we still have good data coverage in the West. So that's great. So in the data set that I've generated, we have, so the CQ data, and then we also have watershed features. Okay, so where is it? So this QC attrib, those are these are the watersheds that have been, these are all the watersheds that have our data. And I also kind of included a bunch of attributes, so watershed attributes. So we're looking at urban land use, natural land use, agricultural land use. We can see here we have fertilizer inputs. So it's a magnitude of how much nitrogen fertilizer is going into every watershed. And that's an estimate. And it's also temporal data. We have manure, how much manure is going into every watershed over time. BFI, that's base flow index. So this is the fraction of the stream flow that can be attributed to kind of the groundwater flow. So essentially, if it's high, it means that you have a lot of your river water is going to be from groundwater. If it's really low, then you see most of it coming from kind of runoff and less from groundwater. So if we're trying to understand maybe the impact of groundwater on nitrogen concentrations, having that BFI metric is really important. We also have a slope percent. So this is the slope of the entire watershed. AI is aridity index. So that is the fraction of potential evapotranspiration to precipitation. So what essentially that measures is if it's high, if it's above 1, it means that it's a very arid or dry watershed. If it's below 1, it means it's a little bit more of a humid watershed. So we're trying to parse out, a climate feature. We're using aridity index to get some sort of climate signature in our data set. Median groundwater depth, so GWD is mean groundwater depth. Tile drainage, that's again, like I said, it's those giant pipes that are in the subsurface. And those essentially just act to move water really quickly all the way through. And then we have wastewater treatment plant density. So that's how many wastewater treatment plants we have in our watershed. So again, if these are unclear, check out the metadata. I summarized them all for you in that. And essentially what we want to, our first approach is really just to start looking at the data. So, you can use this describe function. It'll give you the mean, median, everything else. And those are nice, but I always find it's nice to have a visual of your data. So, what I like to use is histograms. It gives you an idea of how your data is distributed, and also box plots, because it'll give you the metrics that you need, like median, and then the 75th and 25th interquartile. We can see here that watershed area is log normal distributed. So keeping that in mind, if we ever want to normalize the data, that would be something to consider. We of course have the log, we can log it and the distributions become normal. So I think in your workbook, or in your workbook, you have the next step is to look at, let me find it here, sorry, to plot the different land use percentages. For the sake of time, I might, so I included most of the code in there for you. I probably, just for the sake of time to be able to kind of get to the more interesting things. You're welcome to do this if you're fast. If not, we can just kind of continue on my workbook. So, extremely simple. Just we're just aiming to kind of create histograms and box plots for every attribute. And again, this is a little bit of a kind of slow or kind of like it feels really meaningless, but understanding your data and building some kind of gut feeling about it and intuition about it will help you later in the process. So we have our urban land use percentage, percentage of the watershed, agricultural land use, and natural land use. So this is of all the watersheds. So what we can see is that, unsurprisingly, there's not a ton of urban watersheds. That makes sense because urban areas are really small compared to the larger area. There's also a large number of watersheds that don't have any agricultural land use. But there's kind of like an even distribution. So, if we're trying to think about what we want to do with our data, great, we have some agricultural land use, we have some urban land use, and we also have a good amount of natural land use. So starting to kind of think, okay, if I want to look at the impacts of land use on that CQ relationship, this would be a good data set to do that. Okay, so then next we have our, you know, we want to plot urban land use and wastewater treatment plant density. So intuitively, right, maybe, I guess, intuitively, would you expect wastewater treatment plant densities to be highly correlated with urban land use? And I guess that's a question for the chat, if you don't mind just putting it in. Yeah, so I think, again, you know, I think that you think that wastewater treatment plants are associated with urban areas. So, you know, we would anticipate that they would be correlated. And this is, again, just why kind of building that data intuition is important, because what we see is it actually, you know, I think in some ways the distributions don't always look the same. Yeah, and it depends on other variables, right? And of course, you know, wastewater treatment plant density does not necessarily measure the magnitude. We might see more wastewater treatment plants in rural areas because they're a lot more, there's a lot more space between them. So if you're looking for wastewater treatment plant signatures versus urban area signatures, they're measuring different things, even though intuitively they might think that they're measuring the same thing. We're just going to get through some of this, and then we'll try to take a pause on some of the more kind of interesting data exploration pieces. You know, we're plotting fertilizer, manure, and then tile drainage. You know, just trying to get, again, a gut feeling. A lot of our data is log normally distributed. So understanding that when you're trying to make, when you're kind of either asking questions or trying to kind of build models around this, knowing how your data is distributed is really key. So when we, again, base flow index, slope, log normally distributed, and, you know, we're just trying to get these distributions. Okay, so now I have a pairwise plot here, and I have a few questions. Try not to get too far. So take a second, run it, and tell me, what do you see with the data? So I'm going to take a few minutes just to pause. Take a look at the pairwise plot. I'll try to see if I can zoom out so those who aren't coding themselves can still kind of maybe take some guesses at what they see. These pairwise plots are extremely handy, but they could be a little hefty to deal with. So yeah, so maybe I'll just scroll this down a bit. Hopefully you can see most of it, and then we'll move on to the correlation plot as well. You should be able to open the answer key also in, and everything should be run, everything should be loaded already. So if you need to, if you want to look on your own screen, you could also do that. So my question here is, what kind of look at the data, look at how they're correlated, and maybe just give me a few points about what you see in what kind of variable relationships you see. Yeah, so we definitely see kind of three broad types: positive, negative, and no correlation. And so what are some relationships that we see between maybe like positively correlated, but you know, and then kind of with an idea of what you think the variables mean, why do you think they're related? Maybe if you need an example, let's look at fertilizer and manure, or tile drainage. So that's TD, PCT. Right, so we have, I'm not sure if you can see my cursor. But okay, so we see that, you know, we see that agricultural land use and tile drainage are correlated, but not strongly. Okay, so like I said, tile drainage is something that we put in agricultural land, but they're not highly correlated, right? So manure fertilizer seems not very correlated. So going to agricultural land use and tile drainage being correlated, but not very strongly. what we might be able to say, or like what something that might give us something to explore is, oh, sorry, I just lost one of my screens here, my bad. What we might be able to see is, or what we might be able to start asking is, why are we seeing that pattern? What's the CQ relationship between, like, what are the different relations? What are the CQ? How does CQ relationships differ in land use with agricultural drainage or agricultural land use with drainage and without drainage, right? Anything else that sparks any questions? Okay, so aridity index, groundwater depth, and wastewater treatment plant density are not highly correlated. Fertilizer and manure those are correlated with wastewater plant density. So these are all kind of things that are kind of interesting, and again, should start kind of getting your wheels spinning of what kind of questions you might be able to ask. Again, we're just and then so moving on to, let's look at flow and concentrations, understanding the distributions of those data. We see that kind of flow. I transformed it into log flow rate, because log flow rates are often log normal. Concentrations as well. So just kind of, again, looking at how those are distributed. Okay, so we're going to, so now this step is, where are the highest concentrations? We'll dig down here. What do we see? Right, so if you run that, we could run it for different areas. So let's look at the Texas region. So, in Texas, what we end up seeing is that there are quite a few watersheds in dark red. So I guess whoever is from Texas, can we maybe chat about why you might be seeing this, if anyone's familiar with kind of what happens in Texas? And if you're looking from if you're coming from another spot, Western US or something, maybe just comment about what you see, and drivers of what's maybe causing that. And it's okay if it's not, if you don't know the answer. It's, we're just kind of looking at what's happening right now. We're just doing some data exploration.
Danica?
Yeah.
So I had one question. In all of these plots, what do you think? What is the role? Because initially you said the number of data points differs. So does that play a role in creating these inferences? Could that be possible that, maybe I see in Texas at Trinity River Basin, or maybe Brazos River Basin, if the number of points are different, then it might be giving us a biased view, or am I guessing it wrong?
No, absolutely. Right. I think, yeah, being able to, when you're thinking about what you're trying to infer from the data, being able to kind of know that whether you're looking at something because it's a, what's the word? It's a consequence of the amount of data, the resolution you're finding, that you have, or any of that, then you have to be aware of that, because trying to make inferences based on a data set that's not built for kind of a specific reason can get you in trouble, and also just waste a lot of time. Yeah, so Texas, the Brazos River, I don't know exactly which one that is, but what we end up seeing in Texas, you're absolutely right, is a lot of, yeah, and it's a lot of cattle in Texas. There's in the kind of upper part, there is an extraordinary amount of feedlots there. So yeah, absolutely. And you get a lot of irrigated agriculture too, so which poses kind of different issues to water quality. Yeah, so definitely, unsurprisingly, high agriculture, very likely driving those nitrogen concentrations. But it's also worth noting too that, where in the kind of the north of Texas, where there's a lot of feedlots, concentrations aren't that high, right? So, they're in the 0.6 to maybe 0.1 kind of concentration range, which is actually kind of low. So, you know, something's going on, right? Something is causing it. Right. So thanks, Edward. It's very arid. And so that influences the water cycle. Right. So, we can start thinking about, okay, why, we're seeing very low concentrations in Texas. Let's look at the Midwest, for example, though. Right. So they are all above 1.4, which is kind of the median concentration or the 75th percentile concentration. So in the Midwest, the concentrations, even though there's more ag there, but there's still ag in Texas, but we're seeing much higher concentrations in the Midwest. Kind of strange, but then exactly, Edward, you're thinking the right path. It's arid there. There's different drivers. There's different hydroclimatic drivers. So kind of understanding why are we seeing that pattern. So by this point, I hope, we have, or I should say, there's a lot of watersheds that have low-ish concentrations. 0.5 milligrams of nitrogen per liter is kind of low. It's not super low, but it's not that high. A lot of where it exceeds kind of the median concentrations in the Corn Belt, where there's a lot of ag. But there's a lot of spatial variability. So at this point, we can start thinking about, okay, we explored the data. Let's start thinking about research questions. And this is where I also want to ask, kind of, if there's anything that we saw in the data that is maybe just kind of worth exploring, what would be kind of fun to look at. But kind of doing the step before is really important, because knowing what your data can do, like whether that's data availability, like, if we kind of if you don't have many data points in certain area, we might not be able to infer certain things. Knowing how your data is, what kind of data you're working with, can help you kind of generate not just any questions, but the right kind of questions, and the questions that your data set can actually answer. And generally, this doesn't really follow the traditional hypothesis testing approach. It's kind of like play around with your data and then ask questions, instead of asking questions and then finding the data to kind of answer those questions. And I think that's a really important thing that we don't really talk about, is that a lot of the data kind of driven research is not really hypothesis driven. It can be, but you can't really go into kind of hypothesis testing without knowing what data is available, or what data you can get your hands on. Okay, so summary of the data. So I'll just kind of summarize, and then I'm going to ask you guys some things that we could maybe look into. So we have good spatial coverage, fantastic. Land use is predominantly natural. There is quite a bit of natural, but we also have many sites of urban and agricultural land use. So we can kind of ask land use questions. We have other variables, groundwater depth, aridity index, and they have a good spread. So we're seeing, there's a lot of variability in those two metrics across our watersheds. Our areas are like, we have a large range of areas, so we can start answering scale questions. Could be interesting to look at kind of how tile drainage and ag areas are differ. So I have some questions that I wanted that I kind of put together, but I'm wondering if any of you have kind of, having seen the data, kind of maybe some intuition or some kind of your own information that you have based on your own research. Is there anything that kind of speaks to you that might be interesting to explore? Yeah, phosphorus is a whole other bag of problems. Phosphorus doesn't nitrogen is a lot more responsive to hydrology. Phosphorus is less so. So there's absolutely kind of, that's a whole different question. So So one question that we can ask is, how does concentration, just really simply, how does concentration and flow vary across spatial or different temporal scales, right? It's just asking, how does CQ change? How does watershed size influence this? Agricultural inputs, how those influence it? The influence of urbanization on this relationship. How does the water quality trends and concentration relationship trends differ between urban and rural watersheds? So whether that's natural or agricultural. Is there anything else that maybe someone thought of that might be interesting to explore? Yeah, that's absolutely why we see more ag in the Midwest, definitely from the glaciation. Also from kind of really good wetlands, historical wetlands in the area that created really fertile soil. Yeah. So I think, part of the reason why we see lower concentrations or higher concentrations in the Midwest is definitely has to do with lower groundwater residence time. If you could, sorry, can EDA find complex relationships? I mean, yeah, because I think that it's really just about how you're plotting your data and how you're exploring your data. And so I'll jump to the other stuff, and because I would like to get to the end of this, because that's where we actually get to some final data exploration pieces. Okay, so part of it, and I talked about this briefly in the presentation, where we do feature engineering, and this is where you take your existing data and you kind of you calculate new things from it to kind of create new features or just new relationships, and then you could explore using that parameter. So in this case, we're going to use the power law slope. This is a very common metric in CQ relationship analysis. And essentially, what it is the relationship between, if you plot Q, so flow on the x-axis and concentration on the y-axis in log space, the slope that the concentration and the concentration and the CQ data falls on can tell you about, of course, it tells you the relationship between, when flow goes up, when it increases, what does concentration do, and vice versa. So, or not vice versa, but when flow goes, when you have low flow, what's happening with the data, or what's happening with concentration. So, there's kind of three big things we look for. It's the enrichment regime. So if we have a slope that's greater than 0, it's called enrichment. And this is where, as flow increases, so does concentration. So in this case, you can think of it kind of there being excess nitrogen in the landscape, and it's transport limited. So there's just like a ton of nitrogen in the landscape. And so when but it's, it can't really move anywhere because there's nothing moving in. So when flow increases, when there's rain, and then there's a lot more kind of water going through the watershed, and flows increase, this nitrogen that was transport limited at one point can now be moved. So then what we see in this is, as flow increases, so does concentration. We have a constant regime. So that's when B is equal to 0. So as flow increases, concentration remains constant. And so in this scenario, you could think of there just being so much nitrogen in the landscape that no matter how much precipitation comes through, you end up, you're always kind of saturating the kind of the runoff or the water going through the landscape. And so the concentrations just never change, irrespective of how much of what your flow magnitude is. And lastly, there's dilution regimes. So this is where, as flow increases, concentration also decreases. So in this regime, there's usually a fixed mass, like a kind of a fixed mass of nitrogen. So a wastewater treatment plant puts out 5 kilograms of nitrogen per day. And so when flows increase, it dilutes that. So then concentration will go down as flow increases. So in your workbook, you have kind of this portion to fill out. And yeah, so it's mostly there for you. I think I'm just getting you to plug in a few numbers, just kind of the CQ site data, putting the flow in and the concentration. And of course, we're transforming this to log space. So we're going to look at two sites. And so, right. So we'll look at the first site first. Okay. So I pulled up these two sites randomly. So on the left-hand panel, we have the CQ in just kind of like in not log space. And you can see that in this relationship as flow on the x-axis. So, Q is covered there, but on the x-axis, we have flow. On the y-axis, we have C. And what we see is that as flow increases, our concentration increases. And in the log space, we have that positive slope. For our next watershed, we have, as flow increases, we see concentrations begin to decrease. And then in the log space, we have a negative slope. Okay, so we're going to calculate this relationship for all of the watersheds. So I'm just running that function. So I just wrote here a function to be able to do it all together. And I'm using, I had mentioned this kind of group by function. So this is where I take, so the CQ attribute, this is our data frame with all the data. And we're saying group by, so take all the site data. And so like for every site, we have multiple data points, we have multiple CQ measurements. And I want you to take those for every site, run it through the power law function that I just ran on top here, and then spit out what that B slope is. And then we'll summarize all of the, we'll essentially take all of the features that we have, and we just want to put them in the mean. So what we're trying to do here, or I'm just trying to summarize the data. We have the B value, and we're going to take kind of an average of all the features that we have. So then we can kind of start plotting kind of how this B value relates to different attributes. Okay, so let's start by looking at the spatial distribution of the B metric. Okay, so can someone tell me what they see and kind of general patterns? And then if you want to kind of take a stab at why you're seeing kind of the spatial aggregation of what that might be telling you about the landscape, please feel free. Okay, well, so I guess, I think for the sake of time, we'll move on to the next part. So what we see is that enrichment, so positive B slope, very common in the Midwest. Outside of this region, you see more dilution or kind of zero, B slope of zero. So doing another round of, at the start where we look at kind of like how does the B metric relate to watershed properties, kind of understanding those patterns together. That would be a nice iteration to do, but for the sake of time and also because that stuff's a little boring, we won't do it. So next we want to kind of look at how the different attributes, right? So we're looking here. So we chose the attribute urban land use. And then we're trying to see how B changes based on this attribute. So again, we're just trying to see how does B influence, how does B change if you kind of plug it in with different parameters. So, I have all the parameters up here, slope percentage, so we can just kind of start looking at how they look. So let's look at aridity index, or let's look at tile drainage, or we'll look at ag land use. Right, so we see that as ag land use gets higher, it kind of narrows into, the B gets closer to 0. So that means that we're getting more of that constant pattern. So this makes sense because there's a lot of nitrogen in ag landscapes. So you're not really getting, no matter how much flow is coming through, there's always more nitrogen available. And if we plot it with percentile drainage, we see that there's a subset of it, but it behaves a little differently. So kind of plotting these things together, you can get a better insight of how, maybe what drives them. But again, it's hard to kind of, you can get through this, you can kind of look at these plots, but this is so multidimensional that sometimes it's useful to have a dimension reduction approach. So for the last few minutes, I'll jump into the last kind of part of this, and this is where using kind of, we can use machine learning not only as like a final step, but actually in our kind of data analysis process. So we're going to first start, like I said, kind of trying to understand all these attributes and the influence on B. Multidimensional, very challenging. So let's reduce the dimension so we can get a better understanding. So first, we're going to normalize the data. And so we're taking all our attributes, we're normalizing it. So I took all the kind of high outliers, capped it, and then we're using a standard linear normalization. So we're isolating the attributes, just saying, okay, drop all like the site name, lat long, all that stuff, drop that. We just want the attributes. And then, so we're just normalizing it here. You might want to take another look at correlation, just make sure your data looks okay after you've normalized it. And we can see that there's a lot of it kind of covers the whole screen. So good, it probably normalized properly. Okay, so we're going to apply k-means clustering, and the one hyperparameter in k-means is the k, how many clusters you're going to have. And to be able to choose this, it's again very much it's an art, not a science. So you can use something called it's a hyperparameter tuning where you can kind of put a bunch of different k values in and see what the error is for kind of your data set. So for the clusters. And then you just kind of pick one. There's kind of approaches to pick it, but again, a little bit more of an art than a science. But looking at this elbow method, looking at the plot here, the sum of square error, so that the sum of square distance kind of starts getting flat around 8. So let's just go with 8 and see what it tells us. Okay, so we're using k-means with 8 clusters, and we're putting all the features in with it. So what are we going to see? Or so I guess this is what we have, and we have the clusters on the last column. So here, what we just do is I'm just trying to melt the data together, get the attributes, and then we're just going to plot what each cluster, the median value of each cluster. So we took all our data, we clustered it up into 8 clusters across the US, and we're saying, what is the median property of each of those clusters? So this is a great way to just reduce the dimensions of it, so you can kind of get a better high-level view of maybe how watersheds are grouping together. So there's a lot of information here, and I summarized what each cluster each cluster might the properties that are seen. And we'll just go through, I think, 2 of my favorites are cluster 0. So you see the purple ones in the Midwest, the dark purple. These ones are heavily tile drained. So if you look at the TDPCT one, you can see that the median tile drainage value for cluster 0 is really high, whereas all the rest of them are really low. So those watersheds they have a B value of the median value is 0.25. So their enrichment, most of them are being enriched. They have high tile drainage and high ag land use. Whereas if we look at cluster 3, there's an urban cluster, cluster 5. So they're all labeled here. We can see that these ones have a high ag land use, and they also have a high wastewater treatment plant density. And these watersheds have a B value below 0. So those are the ones that we're seeing kind of dilution. So I hope what you kind of could see from this is by using something like, just k-means clustering to be able to kind of reduce the complexity of your data, you're able to kind of get closer to seeing insights of what your data might be telling you. And this is not necessarily the last step, right? We see that the B values are kind of all over the place, and they are kind of clustered together. So there's probably a little bit more work that we'd have to do to be able to really tease out what are driving what drives this B metric. But again, this is one way to just help you kind of get your head around how much data you have, to just be able to say, like, oh, we're seeing this pattern because of this and this and this. And so it's just kind of another approach just to facilitate that. So that's the gist of it. I suggest reading through kind of the different clusters. Again, this is posted on my GitHub. You should have it in the folder already. I apologize that it was kind of a little bit of a kind of quick run through. But I definitely encourage you to go through the data kind of cleaning step, and then also kind of maybe turning back to this and seeing what more you can discover in the data. And like I said at the start, we haven't even looked at temporal trends, and I put a few prompts in there in case you want to do more exploration on your own time. And with that, I will turn it back to all of y'all.
Great. First thing first, I have shared the link to the feedback form, so please fill that up. And that was a great presentation, Danica. Maybe we can take a couple of questions before we wrap up.
Yeah,
If that's
absolutely.
okay.
Yeah.
Okay, so the first question is, what's the difference between machine learning and PCA or FA approaches that reduce the number of dimensions in your data set? Would one not get the same thing for both approaches?
Yeah, that's a really good question. And I mean, they are different methods. So PCA is really useful, but you do lose a little bit of explanation because you're grouping things, like, you're taking this multidimensional data and you're grouping into two axes. So you are kind of, you're losing some insight, but there's absolutely applications for PCA. Just like k-means clustering, k-means is a little bit messier, and because you're relying on the algorithm, or like kind of k-means to kind of group watersheds together using a distant metric, sometimes things don't kind of piece out very nicely. So the difference is that they're very much just different approaches. And I suggest using both of them, using multiple approaches to reduce the dimensions, because chances are you'll find kind of different things using different approaches. So I hope that kind of explains it. I guess, PCA is just, again, PCA is again a machine learning approach, because you can use PCA, put it into a regression model, which is, again, just a machine learning model. So it is kind of just different tools for different approaches to kind of maybe get at similar things. Sorry, I hope that was an adequate answer, but it is a little fuzzy because it's kind of there is no direct answer to that.
Yep. Maybe we can take a very quick one here.
Mhm.
So are there recommended open source tutorials or trainings to follow for geospatial EDA?
Yeah,
One I
that's a
can
great
say,
question.
we have a notebook if you want to follow geospatial EDA, and we often do trainings in AGU. So anyways,
I
to
love
you.
that. Yeah, I'm glad you suggested something, because I don't know of any. There are some professors that do have kind of like courses, online courses, open courses that allow you to kind of do some of this. I think usually more in the machine learning. There's not too much data exploration, because it's not this kind of, it's a little bit of a nebulous thing, data exploration. It's like, you're kind of trudging through the forest hoping to find something. So I don't know of that many. I would like to see more though. So sorry, that's a bit of a non-answer as well. But I think there's definitely room for more of these tutorials, especially with geospatial data.
Great. I think it's time, and thank you so much. It was wonderful presentation, and thank you everyone. See you next time.
Yeah, thanks everyone.